Okay, so welcome to this next video in which we are discussing the antithrombotic mechanisms of the endothelium. Okay, so in this video we're now going to actually start looking at the antithrombotic mechanisms of the endothelium. So, how does the endothelium stop the process of thrombosis from occurring? So the first one we're going to talk about is a protein that isn't actually produced by the endothelium, but is instead produced by the liver. Okay, so I will attempt to draw a little liver over here. I suppose it's not that difficult. Okay, so here is the liver, and it's producing a protein known as antithrombin 3. Okay, and I should just say that antithrombin 3, its strict name is antithrombin 3. However, antithrombin 1, 2, and 4 are pretty trivial. Their effects within the body are almost non-existent. So antithrombin 1, I think, may well have an effect, but antithrombin 2 and 4 have just been, brushed aside as just trivial. So antithrombin 3 is the only one really which is vastly important. And so if people just talk about antithrombin and they don't put a number afterwards clarifying which one of the four they mean, it's likely, in fact it's more than likely, it's very probable that, very much so probable maybe, that they are talking about antithrombin 3. And by the way, antithrombin 3 is also abbreviated to AT3. Okay, right, so what does antithrombin 3 do? Well, basically, firstly, when it is produced by the liver, it's not active yet. So what has to happen is it has to bind to something on the surface of endothelial cells. So let me draw some endothelial cells here. So these are nice, healthy endothelial cells lining the lumen of a blood vessel, okay, with nice tight junctions in between them. Right, so let me colour in the basement membrane in our turquoise colour again, so that we know what we're looking at. So, basically, on the surface of the endothelial cells, there is a glycosaminoglycan, which is basically a polysaccharide called heparan sulfate. Okay, so on the surface of the endothelial cells, you have a polysaccharide known as heparan sulfate. Okay, and if you would like to know more about what heparan sulfate actually is, I have an entire video on antithrombin 3 and heparin, and in that video I explain what a glycosaminoglycan is, and what the difference between heparan sulfate and the drug heparin actually is. Well, actually, heparin is also an endogenous glycosaminoglycan, so it's probably unfair to call it just a drug, okay, but it is used as a therapy for thrombosis. Okay, so this is a glycosaminoglycan, and basically it's just a polysaccharide, loads of sugars linked together, and it has a lot of sulfate groups stuck off the side of it. Okay, right, so we have this polysaccharide on the surface of our endothelial cells. So here is our polysaccharide on the surface of our endothelial cells here. And basically this polysaccharide has a very high density of negative charge because of all the sulfate groups. So what happens is that antithrombin 3 comes and binds to heparan sulfate. So here is antithrombin 3 coming and binding to the heparan sulfate on the surface of these endothelial cells. And once it binds to the heparan sulfate, it becomes active. Okay, so let me colour in antithrombin 3 in vivid purple here. Now, what does antithrombin 3 do once it has been activated? Well, basically, it has roles in preventing thrombosis, and it works by stopping the coagulation cascade. So you remember how I emphasized how important the formation of the fibrin strands, which make up the fibrin mesh, which holds the platelets together, is in the formation of a thrombus. So if you don't form the fibrin meshwork, then you're not going to form a thrombus. You have to have the fibrin meshwork that holds the platelets together, because the adhesions just between the platelets are too weak to actually hold them together in the face of a powerful blood flow. Okay, so if you're going to form a proper thrombus, then you need fibrin deposition. So antithrombin 3 is going to target the coagulation cascade members and stop coagulation from occurring, and therefore stop the production of fibrin strands, and therefore stop the formation of a viable thrombus. Okay, so antithrombin 3 then, now that it's activated, inhibits a whole bunch of things that are within the coagulation cascade. The main one, though, I think you'll probably be able to guess what the main one is. Look at the name. Basically, the main one it inhibits is thrombin, or factor 2A. So thrombin is also called factor 2A. So it inhibits this enzyme. And remember, thrombin, or factor 2A, is involved in converting fibrinogen into fibrin. Okay, so it was involved in both the intrinsic and extrinsic pathway. Both of these cascades just led to the activation of thrombin, basically, and then thrombin is what's going to convert fibrinogen, this inactive component of the blood, into fibrin, which can then be assembled into fibrin strands. So here is the main target for antithrombin 3. It's going to inhibit factor 2A, okay, and thereby stop coagulation. Okay, and this, is important to note, is in both coagulation cascades. So if you inhibit this, you stop both coagulation cascades. But antithrombin 3 doesn't just stop there, it does more than that. No, it does far more than that. It's sort of other targets which are maybe less important than, well, they are less important than its inhibition of thrombin. But it also targets factor 9 through 12. So factor 9A is also inhibited by antithrombin 3. Factor 10A is also inhibited by antithrombin 3. Factor 11A is inhibited by AT3. And finally, 12A is also inhibited. And I just should mention that all of these enzymes here, they are a type of enzyme known as a serine protease. So these are all serine proteases. They cleave bonds which have been formed by a condensation reaction by adding water back in. So they are serine proteases. Okay, right. So basically, then the antithrombin 3 is then a serine protease inhibitor, and there's a nice little name for molecules which inhibit serine proteases. They're known as serpins. Okay, so you may well hear antithrombin 3 referred to as a serpin because it inhibits serine proteases. So the SER is for serine, the P is for protease, and the IN is for inhibitor. Okay, so the antithrombin 3 is a serpin. So let's highlight on this picture all of the enzymes which are inhibited by antithrombin 3. So we've already put in thrombin, or 2A, but we've also seen that it activates 12A up here, or activated Hageman factor. It also targets 11A here, then 9A, and you can see that this is just targeting basically the entire cascade here, and also 10A. And then in the extrinsic pathway, it then targets 10A as well here. So the end portion of the extrinsic pathway is targeted. So basically, you don't just inhibit thrombin, you inhibit the whole cascade. So this is very, very potent at stopping the coagulation cascade from happening, basically, by, if you do form any of these active enzymes, it will bind to them and inhibit them, basically. Okay, so that's stopping the coagulation cascade from being started in this healthy blood vessel. Okay, now let's move on to the next pathway. Okay, so another thing that the endothelial cells are doing is they also have another protein on their surface. So let me show this. So here's an endothelial cell again. In fact, I should have just extended it on. Never mind. So here's another endothelial cell. And another protein that is on the surface of these endothelial cells is a protein known as thrombomodulin. Okay, so this is thrombomodulin. Thrombomodulin. Okay, so what does thrombomodulin do? Oh dear, that's a long name. It's also often abbreviated to TN, and this will be useful in future pictures. Right, so thrombomodulin binds to thrombin, okay? So thrombin is going to come and bind to thrombomodulin. So let me highlight these in separate colours. So in green, we'll have the protein thrombomodulin. So there's thrombomodulin. And then in red, we'll have thrombin. So if you produce some activated thrombin, or factor 2A within the bloodstream, what will happen is it will bind to thrombomodulin, and now thrombomodulin is going to basically not just bind and sequester this enzyme, but it's actually going to reverse its function. So before, thrombin was going to, when thrombin was free in the bloodstream, it was going to convert fibrinogen into fibrin, and then fibrin was going to be converted into fibrin strands to cause coagulation. Okay, now what's going to happen is once the thrombin has bound to thrombomodulin, its function goes from being pro-coagulation, i.e. in favour of producing coagulation, to being anticoagulative. So it's actually now going to have an anticoagulative effect. So what it's going to do is it's going to act on another component of the blood, okay, another coagulation factor. Now, this is actually coagulation factor 14. So, if my Roman numerals are right, that should be 14. So an X, an I, and a V. So X is 10, and then the IV is 4, so 14. Okay, but you don't usually hear this referred to as factor 14. Instead, its more modern name is to call it protein C. Okay, not to be confused at all with protein kinase C. Okay, so protein C is a normal component of the blood. It is a coagulation factor, and when it's activated by the thrombin bound to thrombomodulin, it's going to be converted into activated protein C. And you could also denote this as 14A. So this is activated protein C, which is often abbreviated to APC. Okay? So everything about this silly protein C is confusing, because APC usually either stands for Adenomatous polyposis coli, an important protein in the Wnt beta-catenin pathway, or it may even stand for anaphase promoting complex, an important protein in the cell cycle, but now here it also stands for activated protein C. So not only was protein C debatable as to whether it could be confused with protein kinase C, now it's confusable with all those other ones as well now. Okay, so maybe it would have been better to just call this factor 14A, then it can't be confused. Okay, right. Now, factor 14A is anticoagulative effectively. So it's now joins another protein. Okay, so this is an enzyme, but it's also got a cofactor. So again, this is an enzyme that needs another little protein to come and stick inside it to actually activate it to function. So here we now have the factor 14A, but you need another protein to come and sit here, a cofactor for this enzyme. So I'll highlight this in blue in order to actually get it to work. So what is the protein that comes and sits and acts as the cofactor for the factor 14A? Well, it's a protein known as protein S. And protein S is produced by endothelial cells. So here's another contribution of the endothelial cells. So they're producing protein S, which is the cofactor for activated protein C. So, to review, when you get some thrombin formed in a healthy blood vessel, the thrombin will bind to thrombomodulin, and the thrombomodulin will just laugh at this thrombin and say, you're not going to have a procoagulative effect, you're going to help me have an anticoagulative effect. So they take protein C, or factor 14, and convert it to activated protein C, or factor 14A, which then requires another cofactor, which is protein S, which is produced by the endothelial cells. And once you've got this activated enzyme, what it's going to do is it's going to degrade factor 5A and factor 8A. So it inactivates two components that are necessary for the intrinsic coagulation cascade. So it's going to inactivate factor 5A and also factor 8A. Okay, which remember were these cofactors for the factor 9A and factor 10A respectively. So 8A was the cofactor for 9A and 5A was the cofactor for 10A. So if you reduce these two, so these are the two targets for protein C, protein S complex, 8A and 5A, and if you remove them, if you completely remove them, then you're going to stop the coagulation process. So usually thrombin would have an effect of increasing the levels of these, but when it binds to thrombomodulin, its overall effect is going to be to decrease 5A and 8A. Okay, so we'll continue this discussion in the next video, where we'll see more mechanisms that the endothelium has for preventing thrombosis.